Okay, now we'll move over to plant culture, and we're going to start by just looking at plant tissue culture. And plant tissue culture is really marvelous. So these are plants growing, and this is all in exenic culture. These are baby food jars, and they are growing at the bottom of these baby food jars. And you can see what happens is they form a, that's not very big, they form a callus on the bottom, and the callus looks very much like a cancer growth. And so what you start with is just a small piece of tissue. Here, let's go ahead and look at this one right here. That doesn't make it any bigger, so we'll just start there. So you take you start off with a plant, and you go ahead and excise a piece of tissue. You have to sterilize it first, and when you sterilize it just in normal bleach, with Clorox bleach, 10%, and then you rinse it several times with water, and let's see if we can. So 10% bleach, and you don't have. Well, I don't have a picture for it, but anyway, let's go back here. So you sterilize this, and then you put that piece onto agar medium, which contains all the micronutrients. We even give it sucrose, so they don't have to do photosynthesis if they don't want to. You put it on a 12-hour light cycle, because that's important for them to just regulate their, to maintain themselves, not necessarily for photosynthesis. And then you also add hormones. And we have two different classes of hormones. We have auxins and also cytokines. And we can quickly look at that. Cytokinins, pardon me. See, I'm so used to biomedical science that I forget what cytokinins are. It's not cytokines, but cytokinins. And so auxins, what they tend to do is they tend to favor rooting in plants, whereas cytokinins tend to oppose root development, but instead favor shoot development. So if you want to get shoots, you're going to add a lot of cytokinins and just a little bit of auxins. It's kind of a balance. And these are just organic chemicals. And you can add them before you autoclave even. You can put it right into the medium. They're very sturdy. And then if you add an equal amount of auxins and cytokinins, typically you'll get something we call callus. In mammalian terms, you'd probably call it cancer. It's just a big glob of stuff. And so let's look at this here. Well, here's an example of auxins and cytokinins. So here we've added too many of the auxins, so we're getting mostly rooting. Here we've added too much of the cytokinins, we're getting mostly shoots. And so that's very typical. So you just vary that, and it works great. So what you do then is you go ahead and excise, and then you put it onto here with the auxins and cytokinins. And then from there you're going to get rooting. Well, first thing you're going to get shooting. And so we start off here, and you're going to get shoots developing. And then once you get those shoots developing, then you transfer them. Here's some grass shoots, some wheat probably. And you transfer those shoots over to medium, which contains the auxins, and then you get the shooting. Okay? So a leaf piece, and then callus, and then shooting, and then rooting. Okay? And that's basically how tissue culture works. Very inexpensive, very easy. Some plants do it really well. Other plants do it just terribly. So it really depends on the plant, and it takes a lot of work to optimize the system. Okay, let's go ahead and look at Agrobacterium next. And this is going to allow us to transform plants. And we apply the Agrobacterium in the beginning of the plant tissue culture process, and we're able to transform plants. So Agro has the miraculous capacity to, and we covered Agrobacterium, didn't we? No, we didn't. No, I guess we haven't covered it yet. But Agrobacterium has the amazing property of being able to excitable up to a plant cell, and it secretes a portion of its DNA into the plant cell. So here we have the TI plasmid. TI stands for tumor induction. Tumor induction. And then we have tDNA. tDNA stands for transfer DNA. And this TI plasmid is quite large. It's about 200 kilobases long. And the tDNA, transfer DNA, pops out of this. It is secreted out of the bacterium. It is introduced into the plant cell. This is all with proteins provided by the bacterium. It makes its way to the nucleus, and then these proteins will cut it, and you can see here we have left border, right border, just like a retrovirus. This thing cuts it and inserts it into the plant genome. And this only occurs with Agrobacterium and with plants. It occurs in no other place in all of biology. And so when that occurs, you can have a transgenic plant. So if we look closely at a TI plasmid, you can see we have an origin of replication for replication in Agrobacterium tumefaciens. And then we have this virulence region. And the virulence region codes for the proteins which are responsible for transferring the tDNA out of the bacterial cell into the plant cell and into the plant nucleus and into the plant DNA. So, among other things, that's what the virulence genes code for. And then this is the tDNA region, and there's a left border and a right border. And here we have genes which stimulate auxin production in the plant. And here we have genes which stimulate cytokinin production. And so when the Agrobacterium invades the plant, it causes it to form a cancer. It causes it to form a callus or a gall in the plant. And when that happens, the Agrobacterium is able to sit in that gall and live a life of protection in that gall as a pathogen. It also has this opine synthesis, these opine synthesis genes, and opines are amino acids such as nopoline and octopine, and they are indigestible by all organisms except for Agrobacterium. And Agrobacterium has special opine catabolism genes in the TI plasmid. So let's look at this. This is the tDNA. That's the part that leaves the Agrobacterium and goes into the plant. So the plant has this. Instructions to form a gall and instructions to form opines. Remaining in the bacterium is this. Virulence regions, which allow for this to transport, and opine catabolism genes. So the opine synthesis genes are in the plant cell, the opine catabolism genes are in the bacterium, and that way the bacterium can digest all the opines which are produced. No other pathogen can get into the plant because they can't eat any of the opines. So it's a really nice system that the bacterium makes. And when you look at this, Agrobacterium will put galls. And this is what normally happens, and you can see all these amazing galls here. It's just really something. This is what Agrobacterium does. And so here's a gall, even on a city street, and Agrobacterium made that gall. There's Agrobacterium rhizogenus, which forms galls in the roots, and this is the Agrobacterium itself. So all these galls form naturally in nature, and that's what its gig is in nature. But if we go and look at what we're looking at here, some very clever scientists said, hey, I wonder what would happen if we took all this stuff out and we put a kanamycin resistance gene in there, just like we saw before, you know, right? For the knockout cells. Well, that's a clever idea. Well, they did it, and then they selected on kanamycin, and by golly, they got transgenic plants with a kanamycin resistance gene in there. And so, you know, next step up is someone said, I wonder if there's something commercially viable we could put in this. And they did it, and they put in a resistance gene against insects, the cry genes from Bacillus thuringiensis, and they got caterpillar-resistant plants. And then and they did this at Monsanto, and then at Monsanto they said, we've got this fantastic new herbicide we developed called Roundup, and it's environmentally very safe. It degrades within 2 weeks. It's a fantastic chemical. And why don't we, we've got Roundup resistance genes from petunia and from bacteria, why don't we put them in the plants? And they did, and they got Roundup resistant plants. So the plants you see genetically engineered, oh golly, about 90% of them, 80% of them have these genes in them. The cry genes and the Roundup resistance genes. And they made a lot of money off of that, but they also did a lot of environmental good doing that. So that's what they did. So that's how you make this. You put a kanamycin resistance gene in there, and then you put your gene of interest, and you get rid of all these things, and that's exactly how it's done. All right, let's see if we have any other nice pictures here. Yeah, this just shows the tDNA going over, and we have the Vir genes helping them out. We have a left border, right border. We've got a promoter, a gene of interest, and a terminator. And then, of course, we also have kanamycin resistance. And nothing else of interest there. That's pretty much it. It's a simple, simple system. And you saw what we were doing with the plant with the plant tissue culture. All you need is an autoclave, cheap compounds, auxins and cytokines are cheap, some agar, some sucrose, some micronutrients, and away you go. And you get stuff like this. You know, you have a little callus, you have shoots developing, you grow out roots. It's just very wonderful, great system. Okay, that's it. Any questions before we leave? Agrobacteria-mediated transformation. That's it. Pretty straightforward. Okay. And let's move on to biolistics.